Okay, so today we're going to briefly cover prions. Prions, these are essentially proteins that have not folded correctly, and then they can aggregate together in clumps and cause damage to surrounding cells. So if you remember, we talked about how protein structure develops, right? We had 4 levels. We had, primary, secondary, tertiary, quaternary, and recall that a lot of the interactions when these proteins are making their unique conformations occurs through hydrogen bonding between the backbones, right? And the side chains or the R groups can either be positive, negative. These might attract each other. You might have some that are polar, non-polar, repel based off of that, acidic, basic, etc. So all these unique interactions of all the side chains on each, let me draw this out, on each amino acid, right? Let's just say you have a series of amino acids with all these R groups that all have different properties. These will all interact and make the protein fold in this unique structure. So in prions, these structures have somehow folded incorrectly. So you can see on the left, we have this correct protein. On the right, the protein's all messed up because it started folding weirdly. So these are infectious agents, but they're not alive. Yet they can still act like they're living infectious entities. And we'll see that in a second. So essentially misfolded proteins, when they come into contact with other proteins that are normal, they can induce the normal proteins to become misfolded as well. So it'll just cause this chain reaction. It's called seeding. It'll cause this chain reaction of proteins that keep interacting with normal proteins and make them misfold. So the main thing is that these proteins are forming aggregates or clumps within the cells and just within your body. So these are most well known for causing what are called transmissible spongiform encephalopathies, or TSEs. So these are diseases that are caused by these prions, which are specific to the brain. So essentially the brain tissue, due to the buildup of all these protein clumps, the brain cells will start dying and deteriorating, leaving these that's why it's called spongiform, because your brain literally becomes a sponge because there's a bunch of holes become forming in your brain tissue. And so obviously this has a lot of neurological effects, can mess with your motor function, and then people usually die within a couple months once they get these diseases. So some examples of TSEs include Creutzfeldt-Jakob's disease, CJD, Kuru, and Scrapie. And actually, very interesting fact about how prions were discovered. The way that they were first discovered was that there was this tribe in New Zealand, I believe, where essentially it was a tribe that still practiced human cannibalism. Yeah, so when people died in their community, right, they would eat the people, they would eat their brains. And they noticed, scientists went into this area of this tribe, and they noticed that after the people ate the brains of the other fallen members, they would get these diseases that would cause them to die off quickly and have messed up neurological function. And they discovered that actually the prion diseases were spreading within this community. So when a person would die who had the prion disease, then when someone else ate their brain tissue, those prions would get transferred into the body of the person who ate, and then that person would get the disease, and essentially this prion thing was spreading through all these people because of the cannibalism. So it's actually a very interesting way how this was discovered, in humans at least. And for example, Scrapie, this is one that's specific to sheep. So it's a similar mechanism as in humans, except it's in sheep. And then there's another one called, you might have heard of mad cow disease, right? This is the same idea here. So that would be in cows, but the same overall function, or same mechanism. And then prions, they've been implicated. They're thought to have association with several other neurological diseases, such as Parkinson's and Alzheimer's. So for example, in Alzheimer's, we get what are called amyloid plaques. So you get these aggregations of prions of amyloid beta, and you don't have to know this, I'm just giving you extra information. So amyloid beta proteins, they misfold and start aggregating, forming these clumps that mess with the neuron's ability to transfer information, essentially. Then for Parkinson's, it's a similar thing, except it's a protein called alpha-synuclein, and then they form, again, these aggregates that are called Lewy bodies that are characteristic of Parkinson's patients here. And then we'll pretty much leave it at that because this year's rules don't really cover prions that heavily like last year, so I'm just going to skim over a lot of this information. Just thought it'd be cool to mention a few bonus facts in between. All right, so causes of prions. We actually don't know exactly what's causing these proteins to misfold and these mutations to happen. But we know they have something to do with the gene of the protein itself. Sometimes mutations can occur in that gene that cause misfolding. And like I said before, right, human consumption or blood transfusion, getting infected fluids into your body, right, these are ways that prions can spread from one organism to another organism. So the one prion that's on our a study list for the microbes and agents list at the end of the rule sheet is this major prion protein, or PRP. So we're just going to briefly talk about this. It's a cell surface protein, so it kind of sticks out of the cell membrane like this, and it can undergo misfolding to become an infectious prion, like we discussed. So the normal form, right? It's called. The normal form, like the normal functional form of this protein, is called PRPC. And it's thought to have we're talking about like the completely normal, not disease form of this protein. So the normal protein, right, it's supposed to have a pretty crucial role, or at least we think it does, because it's what we call highly conserved among many organisms. So that means that many organisms, like sheep, cows, a bunch of different animals, humans, like we all have this shared gene for PRPC, which probably means that it has some important function. We don't know exactly what it does, but as we'll mention on the next slide, we kind of know that it's involved with the function of neurons, and it's actually highly expressed within the neuron cells. So has something to do with your nervous system. Yes, and then when PRPC, right, when it misfolds, so you can see this structural differentiation here. PRPC has this weird loopy structure at the end. A PRPSC is the misfolded form, like this. And this PRPC is the thing that goes and forms those aggregates, right? Like a bunch of these PRPSC molecules will clump together and form these aggregates in the cell. And it's basically what we looked at before. These clumps basically cause the surrounding cells to die off, which causes that spongiform encephalopathy, right? The TSEs, where you have all those holes in the brain, and then you have damage to the nervous system. And then we just mentioned this. And then there's one drug that has been shown to have some positive effects called swainsonine. It's a drug, and it inhibits basically in eukaryotes, right? When let's say we have the mRNA, right? We use the ribosome, and then we make a protein. Let's say this is our protein here, right? It needs to go several steps of processing. If you recall, like for example, in the ER and the Golgi, right? Where we can add some things onto this protein, we can cut some things off. Basically, we're trimming it down, we're modifying it so that basically we get this final product that is the functional protein that we want. So basically, this drug will inhibit some of that processing so that when this infectious PRPSC form of the protein is trying to spread around the cell, it'll get inhibited from processing and kind of just get degraded off before it can go infect other cells.